Good evening. Hello. It's really such a great pleasure to see a full room here in our beautiful location here at 19 Washington Square North. This is NYU Abu Dhabi's home here in New York City. My name is Kirsten Sadler-Edepli, and I'm a professor of biology at NYU Abu Dhabi, and I will be your host for this evening. I am a cell biologist, a geneticist, and I'm curious about stem cells, so I'm really looking forward to the discussion tonight. Um, so, since its inception in 2009, the NYUAD Institute in both Abu Dhabi and New York has provided a platform for exchange about the newest developments in research and scholarship across all disciplines. The Institute in New York offers events here at 19 Washington Square North and welcomes the New York community with all the soundtracks to hear experts talk about their research and its societal relevance, technological and cultural impact, and importance for the world at large, ranging from artistic and creative innovations to issues of equality, inclusion, equity, public health, science, environmental sustainability, among many others. As a hub for vibrant, scholarly public engagement, the Institute is unparalleled in the Gulf, and we are pleased to offer programming and extend the Institute's reach here in New York City. This is the first event of the NYUAD Institute of the year, and I welcome you back to any other events that happen throughout the year, throughout the academic year. So I'll start tonight with a little bit of an introduction about tonight's topic, talk about why we are discussing stem cells, why I thought this was a great idea. We'll have some time for questions at the end. And if you don't get to ask your question right at the end, we will have a reception afterwards. And we welcome all of you downstairs to our lobby for a reception right after the event, which should wrap up a little bit after 6 o'clock. So there have been lots of attention given to stem cells over the past many, many years, from things celebrating incredible cures like this diabetic woman who doesn't need insulin anymore thanks to stem cells, this remarkable stem cell therapy for Parkinson's disease. And also, there are commentaries such as this recent one in the New York Times, which asked, are stem cell therapies safe to try? So there's a lot to discuss on the topic, and it comes out in the news and in conversation, but very rarely does it really get to the heart of the science. And that, as a scientist, that's what I really wanted to focus the conversation on, as well as focusing on some of the exciting clinical advances that are happening in stem cell biology. So we're joined here by two real experts in the field. So, we like to have these conversations for the public with people who have spent their lives and their careers thinking about the topics and can really bring substantial expertise to the discussion. So we're very, very lucky to have Dr. Sarah Millar. She is the director of the Institute for Regenerative Medicine. I need to read them all because there are many titles here, Sarah. She and the Black Family Stem Cell Institute. She's the dean for basic science and a professor in the departments of cell, developmental, and regenerative biology, oncological science, and dermatology at the Icahn School of Medicine at Mount Sinai, uptown. She's an extraordinary scientist and a leader. She and her team have made significant advances in their work on stem cells in tissues such as the skin, where they've uncovered fundamental aspects of regeneration with the aim to develop therapeutic approaches to conditions such as skin cancer, hair loss, and wound healing. She's joined by Dr. Lorenz. He's the director for the Center for Stem Cell Biology at the Memorial Sloan-Kettering Cancer Institute. He is a professor and the Haupt Chair in Developmental Biology. His groundbreaking work using stem cells to generate neurons to treat Parkinson's disease earned him a MacArthur Award, so he is a certified genius. And it was featured in one of those news items that I showed you on the first slide. He is really a leader in in the stem cell field, serving as the president-elect for the International Society for Stem Cell Research, which is really the organization that that organizes stem cell research meetings, advises on policy, develops tools and information to for clinicians, for educators, and policymakers. And I put the website up here because I really feel like it's an incredible resource for any of you who are interested in learning more about stem cells and hearing about some of the recent work. So it's really my pleasure to welcome them and all of you to this discussion with a hope that you walk away with an understanding about what stem cells are, how they work, and some of the really exciting advancements in clinical medicine where these cells are coming to bear. So with that, I'd like to start the discussion. Okay, so welcome, Sarah. Welcome, Lorenz. Sarah, I'd like to start with you. So I gave a very brief introduction about you, so I was hoping you could share with us a little bit about you and your work as a stem cell biologist. Also, giving just an overview to the audience about what stem cells are and what model systems are being used to study them.
Oh, thank you, Kristen. Can everybody hear me okay? No? All right, I'll just yell. Okay. How's that? Better. Okay, thank you. So thank you very much for the very nice introduction. Yeah, so my name's Sarah Millar. I'm a developmental biologist and more recently a stem cell biologist. So I really came to science from understanding how embryos develop and how skin develops in particular. But now my lab focuses mostly on postnatal skin and how it's regenerated. I'm originally from England, as you can probably tell, but I've lived here for a very long time. Louder? Okay, there we go. Right up here. Okay. I'll pretend like I'm Madonna or something. Okay. So what are stem cells? So there are many different types of stem cells. The original stem cells are in your embryo, and they give rise to all of the different tissues in the embryo. And so they're known as pluripotent stem cells because they can give rise to all of these different types of other cells and tissues. A very important aspect of stem cells is that they can renew themselves. So they have a very long potency and lifespan. So once you get an adult tissue, we also have stem cells in those adult tissues, such as in the skin, which I study, and hair follicles and intestine and lung. And those stem cells serve to regenerate the tissue throughout life. During aging, these what we call endogenous or innate to the person, the stem cells, they can basically get tired and run out, and that leads to many aspects of aging, including problems with skin and hair loss, which are things that we study in my lab. But there are many other defects as well that occur in aging in part because of the poor functioning of stem cells. So one of the focuses of my lab and of many of the labs at Sinai is to really understand these adult tissue stem cells and how they function and what goes wrong with them in aging, with the idea of identifying new therapies to halt or slow down aging processes. But there's also another whole aspect to stem cell research, which is very exciting. And that's that you can actually take those embryonic stem cells and grow them in a dish and replicate part of the process of embryonic development, but outside of the body, basically. So those embryonic stem cells can be coaxed when they're in a dish into any type of tissue or cell type. So when this was discovered, it was extremely exciting, but there are some ethical considerations around using stem cells from human fetuses, which give some people a lot of pause. So what was even more exciting in a way was when it was discovered by a Nobel Prize winner, Yamanaka, that you can actually take adult cells from tissues such as the skin, and you can reprogram them so that they function almost exactly like embryonic stem cells. And they get reprogrammed. Originally, the way they were reprogrammed was by introducing a set of transcription factors, and those are proteins that activate gene expression. And there's a specific set of these that will allow you to convert a blood cell or a skin cell from an adult person into what we call an induced pluripotent stem cell. So that's super exciting, because now we can basically take from any one of us a small blood draw and make any kind of tissue or cell that we want. So that this has a number of different exciting applications. One of them is that we can use these cells to study disease processes and screen for drugs that could inhibit those processes. And this is really cool because these are human cells, and while animal systems remain extremely important in disease modeling, animals such as mice, which we often use, are not exactly like humans. And so many drugs that will work in a mouse won't necessarily work in a human. So having this human-based system for drug screening is really cool. The other very cool aspect about this is that we can personalize this. So we can take cells from a specific patient and mimic their disease in the dish. So we can screen for therapies that would specifically work in that person, because as we know, there's a lot of individual variation between different people. And then the final thing is that we can take these, because we can take these cells and make them into specific cells or tissues, we can also use them for transplantation for therapeutic purposes, which is something that Lorenz will speak much more about. His lab is doing some really exciting work in this area. So we can, Kirsten already mentioned treatments for Parkinson's disease and for diabetes, but there are many other potential diseases that can potentially be treated in this way. So that's sort of a little bit of an overview of what stem cells are. So in addition to running my own lab, I'm the director of a stem cell institute at Mount Sinai. We have a very large collection of scientists who I'm really privileged to help do their work, basically, as director, and try and bring resources for them. We study in different labs at Mount Sinai pretty much every organ in the body, and all of those organs that rely on stem cells. We're intensively studying them. And we also have a big IPS or induced pluripotent stem cell program with very exciting work focused on Alzheimer's disease, Parkinson's disease, and other neurodevelopmental and neurodegenerative diseases, as well as other areas, including cardiac and lung and tracheal issues. So with that, I think I'd like to pass to Lorenz, who will tell you about incredible work that he's doing.
Thank you, Sarah. So, Lorenz, I'd really like to hear, from you about you and your work, and also if you could talk a little bit about the stem cell community at MSK. So here in New York City, we're really fortunate that there is a very robust stem cell community. I should mention that there is a wonderful stem cell center at NYU, as well as the Stem Cell Institute, and of course at Mount Sinai, Sloan-Kettering. So we're looking forward to hearing from you, Lorenz.
Great. No, it's a real pleasure to be here. I hope you can hear me. The microphone doesn't work. Maybe it does work. Yeah, so as you can hear, I'm also not from the US originally. I'm originally from Switzerland. And it's actually where my journey started, getting interested in stem cells. Because at that time, I was really thinking, how can we think of new regenerative approaches, particularly in the brain. So the brain is a little bit a special place. There is some evidence that you have stem cells throughout your life, but they're quite limited in their function. They might have maybe some function, particularly in animals, for smell, maybe for some memory retention, some memory tasks, but for most other function, actually looks like there are no stem cells left that would replace when a nerve cell is lost. So that raises then the problem. Now, if some of the cells that we are interested in, such as, for example, in Parkinson's disease or in Alzheimer's disease, those are cells that were also derived from stem cells, but they were born when the embryo was still in the womb. So those cells have been born and have survived, in some cases, for 80 years. And obviously, they become vulnerable over time. They might degenerate, particular certain conditions, certain genes that can contribute, certain environmental factors. Happy to discuss that further if someone is interested in. But once they are lost, there is no stem cell in the body to replace them normally. And that really was for me the trick. How can we replace that? And I thought on my side, we had such a nice broad overview from Sarah. I will give you a little bit more kind of a specific aspect of our work that really went throughout my whole career for a good 30 years now, where we actually initially looked what could be the right source of those cells. So for example, we actually, when I was back in Switzerland, we really used fetal stem cells from the brain itself. And we went all the way to grafting them into the brain of Parkinson's patients. But it was so difficult to do that properly, not just that there are ethical concerns or not, beyond that, just practically. It was impossible to get enough cells to do that as a routine technique. And so that really got me on a big journey. Now, what is the right cell? Can we really grow these cells in a dish? And we heard, again, from Sarah, there are a number of stem cell types you could use, not only the ones that are normally in the body retained in adult, but those embryonic-like stem cells are now the ones we can make from any one of you, these induced pluripotent stem cells. So that's for us became the main source. The challenge was really know, how do we get them into these very specific nerve cells to replace? Because the brain has about 100 billion nerve cells. And for example, in Parkinson's disease, the cells that are lost, they are only about 300 to 400,000. So compare 100 billion to 300,000. If you just make randomly nerve cells, you're not going to get the right one. So you need to very specifically know how to make them. And so that's why I'm also, as a chair in developmental biology, we needed to understand how does development do it and then do that in a dish. So that took a long time. It was a lot of basic work. And we still continue to refine, but we basically understand the logic now, how to do that. So that raises in the question, what can you do with those cells? And obviously coming back to my original plan of Parkinson's disease, already started in Switzerland, now with the fetal cells. Can we now do that with the embryonic or with the induced pluripotent stem cells? And so over time, now we made some of the initial breakthroughs that really could show that we can inject those nerve cells, generated completely in a petri dish. And the power of those cells is you can make unlimited numbers. So it's kind of a very unusual cell. Now it renews, but it seems to not age at that pluripotent stage. So you can literally make billions or quadrillions of those cells and then make them into the right nerve cells. And so about maybe 15 years ago, we finally had kind of a breakthrough study that showed you can do that in a mouse, you can do that in a Parkinsonian rat, or you can do it in a Parkinsonian monkey. So that was really exciting. And we're all gung-ho, say, oh, now we're going to go to patients. But it turns out that at least another 10 or 12 years before we actually are doing that. And so that's kind of one of the messages. No, this is a very serious process, and it's quite a big responsibility if you actually want to put these cells into patient. You obviously don't want to cause a tumor or have the wrong cells there. You don't want to cause seizures or something else that these nerve cells could do. You need to have the exact right cell types. So we had to work with the regulatory agencies, with the FDA, with many colleagues, multidisciplinary clinicians, with patient advocates to learn what's really important for the patient that we should restore. And that was really a long journey. But eventually in about 2021, we were finally at the stage that we could transplant the very first patient using this technique, giving them new nerve cells. And it was really exciting because for the first time now, it's not only in a degenerative disease such as Parkinson's, you think the disease is progressive, it gets worse and worse and worse. So people tried to do is they try to slow down the rate of progression. So it gets a bit slower worse, but in this case, they actually try to get new function back. Nerve cells are already gone, we get new ones back, and they seem to integrate. into the brain. So that's really an exciting possibility. And so now we just published actually a couple of months ago, I think you had the paper up, a paper that showed the results now 2 years out, and it looks still quite promising. Now we have to be very careful because it's only 12 patients so far that got this therapy, and 5 got a lower dose, 7 got a higher dose. But it looks like not only was it safe, so now some of those patients are 3 or 3.5 years out, nothing bad happened, but also they seem to actually get better in some of the features, some of the way the neurologist measures how severe Parkinson's disease is. These were patients that even with best medication, they have about 5 hours in the day where they cannot properly move at all. They're like frozen. And so in that case, we could cut in half about that period. It was not a complete cure, but about halving those phases. And that's just one of those measures. So it's quite exciting, and led then in collaboration again with the regulatory agencies to now planning of an actually big trial. So when you do clinical trial, it goes step by step. You do a phase 1, phase 2, phase 3. And we're actually allowed to jump from a phase 1 safety study directly to a phase 3 study. And so again, because the results look promising, it's going to be 100 patients starting this year. They're going to be tested in such a way that if the results look promising, this could be kind of a more routine therapy. Now, it's not super routine because it's not just a drug you take and everything is better. You need a neurosurgeon that puts these cells into the right place. So it's still an invasive therapy, but potentially a regenerative therapy. So this was really a long journey. And I think on this long journey, we also learned a lot of, again, what are the challenges and how to do it right, and how maybe some things people try to skip steps. There's always a lot of competition among scientists, among people who have financial interests. I think one thing interesting to discuss there also is really the question, what happens in general? Now, you hear a lot about stem cells. When you do Google searches or Bing search, whatever you do, often you look for stem cell, and you get actually all kinds of ads from companies that sell you stem cells. Some tell you can treat autism, some tell you can treat Parkinson's disease, some tell you can treat all kind of joint disorders and so forth. And typically these are so-called endogenous cells coming either from the blood, from the bone marrow, or from the fat. When you do a liposuction, you have some cells which are called mesenchymal-like stem cells, and they are then just isolated and re-injected. And there might be some interesting applications for those cells. They might have some immune functions, or they might change a little bit how the immune system acts, which can be beneficial. But they're clearly not cells that are going to make new nerve cells. And so that's really a big problem in the field, because the companies, they're going to promise it to you. For a while, they were not allowed to do it in the US, which might actually now change under our new government. But you have to go to Mexico, you have to go to other places, and you have very little information. So that's one of the tasks I actually talked about at ISSCR. We try to help educating patients, not to tell them you have to do this or that, but to give them the information that they can talk to such companies, that they know what is right, what's not right. For example, if you have a therapy that was never approved, which is the case for those therapies, you should be part of a trial. You should not have to pay for this therapy. You actually help developing it. This company is just going to charge you. And so that's already one sign that probably something is a little bit wrong in that context. And again, happy to discuss that in much more detail, but that's one of the big issues I think that is plagued the stem cell field. I think that's going to remain very, very high on the agenda, because it can not only not work. There were some examples a couple of years ago, for example, where they were used for injecting into the eye. In elderly people, there's something called macular degeneration, where you start becoming more and more blind. You can no longer see focused. So they injected those stem cells, now these mesenchymal cells coming from fat and so forth. Not only did it not work, actually the patient got completely blind, because they got a scar-like formation, and it was basically a disaster. And so it is really a big problem currently to address that properly. And again, that's something which ISSCR does, and I'm very happy to also discuss about other aspects. Now, we mentioned some of the ethical questions of getting the right cells. It's less of a problem with those induced pluripotent stem cells, but sometimes people also use embryonic stem cells, and again, there are pros and cons for doing that. And I think there are many important topics. Hopefully we get a chance to touch on this a little bit in this discussion.
Yes, thank you so much for that. I think both of you gave a great overview and some examples. I want to touch on something that both of you mentioned. Lorenz, you said that you really had to go back to the embryo and understand how does the embryo develop and how do these cells develop and differentiate in the embryo. That's a topic near and dear to my heart because that's what my lab studies. But I think that you bring up a really important point about how it's really that basic science and the understanding of the cell biology, the genetics, the embryology, the developmental biology that is paves the way for then something that you could then do in a dish. And I wondered if you could each just briefly sort of bring up an example or touch a little bit about that on how that basic science research really has been critical for your work or for an example. Either one of you can take that to, yeah.
Yeah, thank you. That's a really wonderful point. And I also think that Lorenz brought up, the time and care that it takes to develop a therapy that's safe for patients. And I think that's something we really have to, everyone's impatient for the next cure, but in order to do it properly, it really takes a lot of time and very careful, controlled studies. So this is an incredibly important point. But to get to the developmental biology, I mean, my lab actually has studied for many years a family of genes that has a very strange name. It's WNT. We call it WNT. And it turns out that these genes are very, very important in many, many aspects of disease and health. But they were originally discovered in fruit flies. And people, sometimes people kind of denigrate the basic science that goes on using model systems such as fruit flies and zebrafish. But that was where this family was first identified. And in fact, many of the mechanisms by which these proteins work were figured out in fruit flies. So it's just one example of how this very basic research that's really driven by scientists' curiosity leads to these discoveries that have huge impacts for human health. And I think that's something that as scientists we don't convey very well to the public often, and it results in kind of not really a great understanding of how the scientific process works and how this kind of curiosity driven, sometimes very esoteric sounding research is what brings the big new idea to the fore.
That's a great example. Thank you. I think we have some Drosophila biologists in the room, so I'm sure they're happy to hear that. Yeah, Lorenz.
Yeah, maybe just to follow up, I mean, we have not only also the WNT pathway, that is very important. When I talked about how do you make all the different nerve cells or all the cell types of the body, it really is like a developmental language. So some people think of programming, the cells, that initially is nearly like a blank slate with regard to developmental information. You can say, okay, there is unlimited number of signaling pathways in the body. How are you going to ever figure it out? But actually it turns out it's only about 7 or 8 of those pathways. Nearly all were discovered in the fruit fly. And you just need to either activate or inhibit the pathway at the right time and in the right combination. So I think of it a bit like a little bit of learning how the body programs the cells into those specific fates so that they are part of the right region and so forth. So it is really a language that is very basic and very fundamental and seems to work across animal systems in many, many ways. Maybe the other example there for me is the fact we are also very interested in the question, not only making the right cell, but making the cell at the right stage. So for example, you want to study Alzheimer's disease in a dish. You want to have a cell that behaves similar to, let's say, a 50-year-old or a 60 or 70-year-old cell, but they're embryonic derived. They're actually fetal-like. And so then fundamentally it's the question of how do you study that? And for us, it was very important to figure out. So actually, other organisms, let's say even the mouse, many people know mice, they also make a brain. And they run around, but it takes them about 20 days to do so. In human, it takes 9 months. So why does it take so much longer in a human system? And so you can do a lot in those systems where you actually go forth and back between species, try to figure out why does the clock seem to move faster in a mouse than a human? Because if you understand that, maybe we can control timing more generally. We already talked about aging, so that'd be nice if we could control that. But also for what we're doing for regenerating the brain, when you put the cells in, they are embryonic, fetal-like, maybe good because they are young and fresh. They can survive in this diseased environment. but they need to mature. And so the patient, to get the benefit, I always say the patient has to be patient, because it takes about a year. This does have to get mature enough. And each cell eventually becomes about as long that it has about 2 yards of connections, a single cell. So that's why I need so few. They become huge. But so it takes a very long time. So if you understood how to make that go much faster, that would obviously help these therapies to be even much more quick. And so I think, again, that's where we can learn a lot going across different species. A lot of basic mechanisms that are going to be very, very essential to have huge impact on aspects not only of regeneration, but again, for example, aging.
Yeah, those are great examples. Thank you. So I know that we're all very excited to hear about the clinical applications. We've talked about some with Parkinson's. I talked about diabetes. There's been a lot in the news about diabetes and stem cell treatment of that. And I wondered if you could just share with our audience something that maybe from the centers that you direct, or your colleagues, or the meetings you go to, something that really is exciting to you. What do you think just sparks your curiosity and your enthusiasm in the clinical application realm? Either one of you can go. Yeah.
I mean, there are a number of areas now that are either already showing that they might work. And so one example that maybe not many people have heard in the brain is actually treating seizures. So there's some really interesting data that quite a common form of seizures, they happen in a specific brain region. And it affects about 100 to 200,000 people in the US. So it's not a super rare condition, and they are treatment resistant. And there have been some really interesting data recently where colleagues in California, actually, they made so-called interneurons using actually protocols we developed a long time ago, and making those interneurons and injecting them into that brain. And because seizure is too much activity in the brain, they can, by inhibiting this inhibitory neurons, adding new inhibitory neurons, that seems to help in toning down those seizures. And so that's something that could be quite powerful, and something that probably most people haven't really heard about, and something which actually now goes into also phase 3 trial. So it could become a routine treatment. And the other area, which is much more kind of pie in the sky, I think, but which we are very interested in pursuing in my lab actually, is to replace not the nerve cells in the brain, but the support, the glial cells. And there is one cell type called the microglia, because they're kind of smallish, and they're very important for the immune function. So if you have an infection in the brain, they would go and try to eliminate those. But it turns out when you look in Alzheimer's disease, most of the genes that are linked to Alzheimer's disease that put you at a bigger risk, there's specific genes expressed in those microglia cells, which is a big surprise. You'd think it's a neuronal disease, neurons go wrong, but it seems to be actually a lot of the genes are in those microglia. And what we and others can do, there are tricks with certain drugs where you can literally swap all the microglia, at least in a mouse. This study is now done in a human, where you can give a new complement of microglia in that brain, and you can make them super microglia. No, not only put them normally back, but you can put in genes that actually are protecting from Alzheimer's disease. And again, it's still an unproven approach where that's going to truly work, but it could be quite revolutionary if it does work, because it would be a method that you can deliver. These cells seem to spread, and a method that really could ultimately have a big impact for the disease that you have to be done at the right stage. No, Alzheimer's disease, if you're very end stage, it's too late. But at an early stage, early cognitive loss, this could be quite a powerful approach. And I'm very excited thinking how that will actually turn out in the long term.
Wonderful. Sarah, maybe you could give us one example, something that's very excited about.
Well, I guess I have to talk about the skin, because
Yes,
that's my
please.
thing. So there's actually a very exciting study that's done a number of years ago to basically cure a young boy who had a genetic blistering disease of the skin that was ultimately going to be lethal, because if your skin kind of peels off all over your body, you end up dehydrating or getting horrible infections. So because they knew the gene that was defective in this boy, they were able to basically introduce a corrected copy of it into skin stem cells and then graft them. And this took over, I think, about a year, but they gradually grafted each part of this little boy's body with corrected cells and basically cured his disease and allowed him to live a pretty normal life. So that was a very exciting example and something that we are trying to replicate for several different types of genetic skin diseases.
Yeah, that's quite a dramatic example. Thank you for that. So, you know, I think that the promise for stem cell, I think we all think the promise for stem cell therapies, the excitement of stem cell biology is really central to our field. But of course, there are ethical considerations and there are challenges. I think, Lorenz, you mentioned some of these about untested products and people who are seeking a quick fix. And so I wondered, Lorenz, if you could speak a little bit about the ISSCR, the role of this society in advising policy, and maybe touch on some of the challenges with using stem cells for clinical applications.
Sure. That is, again, that was a very important part of the mission of ISSCR, which is an international society. So we try to really engage scientists across the globe, again, to have standards that we cannot enforce them. No, we're not a legal organization, but just simply by good practice, give them the tools that they know what are the right standards for characterizing the cells, for running clinical trials, and for really getting these products to the clinic and to the patient safely. And this actually just, again, we had the website up. There is in new data, a new updated version of the stem cell guidelines, which are very detailed, that actually have information for patients. It has information for scientists who want to develop a clinical therapy. They can go there and look at the specific application, in which country they are, to get specific regulatory advice. And so we really try to streamline the process, because one issue that obviously I hopefully got across is, even once we had the proof of concept, we had a very nice paper in a top journal, but it took another more than 10 years to get it to the patient. So can we do that shorter? Can we help each other? Did we learn how to do that faster? And so that is actually now one of the new tools from ISSCR where you can do that. Another important mission is to educate not only patients or the scientists, but actually clinicians. You might be surprised how little many clinicians know about stem cells. So they get the information or the question from their patients. They want to know, okay, should I go to Mexico, get those stem cells? And they often don't really have the right tools actually of doing that. And so what ISSCR has done, they actually made a so-called CME credited course. So when patients practice, they need to always show that they stay fresh in the information. They need to do these courses, and they need to do courses that give them credit. So ISSCR made actually such a course where they can get credit, and they learn the newest about stem cells and how to talk to the patient and what is really good science or bad science. I think that's, again, a very important role, that we engage all the stakeholders in this chain. I think that's a very important area. And then a lot of interesting ethical questions now that need to be addressed, not only the ethical questions of the stem cell tourism, but also for people who want to do it right. For example, how do you really approach a patient about the stem cell therapy that's unproven yet? We maybe go through all the right steps, but you can still not tell the patient it's going to help for sure, no? It's an experimental therapy, so you need to have what's called informed consent and very specific rules that need to be independent of the interests of the scientists. And so these are many, many ethical questions that need to be properly addressed, and where ISSCR makes an important point. And there's also a lot of really emerging new technologies that we always need to adjust, and that's the beauty of ISSCR. Now we have scientists from across the globe, across all types of stem cells, and there are, for example, new models which are called advanced stem cell models, that you can make from the embryos or embryonic stem cells, from these induced pluripotent stem cells, not just individual cell type, but structures that look a little bit like an embryo. They're not really true embryo, but they're called embryo models. So it gets a bit more into this question now, what is really good? How long should you be able to grow them? Could they become problematic? What if someone wants to implant them into a woman, which is forbidden based on those rules and guidelines? But there are always no scientists try to push the boundaries, which is a good thing, but they need to also get the feedback on what is really an appropriate way to do that. And one important function again of ISSCR was to establish this ethical oversight board. Nearly every institution has those. They're often called escrows. and it's basically this stem cell research oversight committees where you have scientists, but you also have lawyers, you have ethicists, you have different people from the community that actually look at such a proposal and say, is that really to the benefit of human society? Is that really justified? What should be the boundary conditions? So it's not just scientists just go crazy and do things that might actually become problematic and might actually hold the stem cell field into a very bad light and controversial light. So we try to, I wouldn't call it police, no, but to have clear guidelines that they should follow. And we try to enforce, for when people want to publish their papers, they actually have to say now which guidelines they have followed. And so it becomes more of a standard of the art of how you do stem cell research. I think these are all very, very important functions of ISSCR, and I'm sure that will continue to evolve over the years.
Thank you so much for that. And I think maybe that's a good note to end on, because I would like to take some questions from the audience. But ending on that note of having this be a global community and bringing scientists together from all over the world. There are some conditions here in America that are perhaps different than the UAE, which are perhaps different than England or Switzerland. And so I think that is really a critical part of the conversation. So I'd like to open it up to the audience. I do see that there are some students in the room, so I want to give the first opportunity for the students to have a voice in this conversation. So if any of you are students and would like to ask a question, the microphone will be passed to you. I don't mean to put you on the spot. I'm in my professor role here for a moment. Any students or trainees? But go ahead, yes.
Yeah, this is a question for Lorenz regarding the insertion of stem cells for Parkinson's. That I know for quite a few years that created some problems, that the brain saw them as foreign objects and created tumors around them. And I would like to see if you can clarify. I know that the last round of 10 or 12 patients somehow were trying to overcome that problem, and I don't know if somehow it got a way to get beyond that problem, or is still a fairly significant concern.
No, I would say at this point, obviously we can never say it's absolute no problem, but I think that problem has been largely overcome, and it was not the main problem actually in Parkinson's disease. From all the studies that people did, fetal studies on, there's no clear evidence that anyone developed an actual tumor. There were some other examples in Huntington's disease where there was kind of call it a benign tumor, an overgrowth. The brain got kind of a little bit large, but it was not a classic, not like a glioblastoma or some really severe tumor. So we think that is not as much of a problem, but maybe something you're referring to is what happened with the fetal dopamines that when they were injected, patients got side effects. And those side effects were actually aberrant movement. So when you have Parkinson's disease, I mentioned it, many patients are kind of frozen, particularly without medication. But when they take medication, if let's say you see Michael J. Fox on TV, he's not frozen. He actually has too many movements. No, he makes all kind of, he cannot control the body, which is called having dyskinesia, abnormal movement. And so what happened in some of the patients that got initially those cells from the fetus, they got this dyskinesia. So one simple explanation would be it's like a drug, it's like too much of it, or maybe at the wrong place. They actually by now think it's not exactly that. We think that in those fetal cells, there were some of the wrong nerve cells that can interact with the drug to cause those dyskinesia. And because we know what those wrong cells are, from the stem cells, we can eliminate those. So in all those studies from our lab, there's a study that was published at the same time as ours in Japan. There are groups in Europe. None of them has seen those abnormal movements so far with those human stem cell derived nerve cells. And so, but there are clearly many challenges. Now, we don't think it's going to be a cure of Parkinson's disease. I always say the best possible outcome would be quote, a cure of the movement disorder of Parkinson's disease. So Parkinson's has this problem with movement, which is one of what people mostly know about. And so if you're really lucky, we can nearly cure or very well treat that condition. But they get actually often other problems. At late stage, some patients get cognitive problems. We don't think we're going to be able to treat that with this approach. There we need another approach, maybe the microglia, maybe something else. So it's not going to be a cure for all the symptoms, but the dream is to have it a cure for the movement disorder of Parkinson's disease. And I think the safety, again, we have to touch wood, I guess, but we haven't really seen any major problem in our studies, any other studies. And even in the fetal grafting study, I don't think that was the main concern at the time. Yeah, but it's a very good question.
I see.
I have a question.
I see. Okay, great.
Yeah. What is the role of stem cells in pain management? Millions, if not hundreds of millions of people around the world are suffering from pain, from a variety of diseases that includes arthritis and all the facets of arthritis. And no doctor, no research, no anything is out there to address and solve the pathway that leads to pain in order to find the gate that will close that opening that creates all of this horrible situation for a variety of diseases.
I can try. I wish I could answer that question. From what I know, which is very little about this, we don't really understand enough about the pathways that lead to pain. And I'm not sure that stem cell therapy is the first route that one would go down, in trying to address this question. I think it's really, firstly, a kind of a neuroscience question of understanding which neurons are being activated to make you feel pain, and perhaps trying to, we already have some drugs, but, find more effective drugs to block that. Another very important aspect of pain, I think, is inflammation. So, I personally have like 3 herniated discs, and I know that when I have pain from them, it's because it's not necessarily from the disc problem itself, but more because the nerves are getting impinged on by the herniated disc, and that's causing an inflammatory reaction. And so the most effective treatment for me anyway is to take a big dose of naproxen, which just kind of kills the inflammation. So, pain is quite complicated. And so there are many different aspects to it. But again, I'm ignorant on this topic, and maybe Lorenz can give you a much more educated answer.
I mean, we did a little bit of work on pain neurons. We developed some of the early protocol, how you make a nerve cell that can sense pain. And so actually different ways of pain, or there is heat-related pain, inflammatory pain, and you can actually study that in a dish. And so what's kind of nice about that, you study now that in a human pain nerve cells, because pain perception, some of the receptors are actually different between mouse and human. And so that allowed some of the pharma companies to now doing testing in those human stem cell derived pain neurons. And so again, I'm not completely on top of the field now, which are the newest drugs that came out from that, but I know they developed some specific channel antagonists that came from those stem cell model. There's also really interesting work from a colleague of mine at Stanford, Sergio Pasca, who tries to build a whole pain circuit. So not only making the pain neurons that we generated, but then he makes the tracts that go into the brain, and even the cells that sense the pain in the brain. And so you can actually then stimulate those neurons and see activation in his brain in a dish. I mean, and you shouldn't call it brain in a dish because it's not a brain. No, it's just small pieces of it. But it's kind of an interesting part because you can at least see the whole circuit. You could ask, so where are things happening? Now, if I have an inflammatory stimulus, which part is affected, which drug acts on which cell? So there's a lot of potential using the stem cells as a model, primarily for studying pain, less so for the treatment. Even though I know some companies that try to do that. Some people tried to use interneurons. They claimed in the spinal cord pain that maybe there's improvement, but I wouldn't bet my money on it yet. And then also a former postdoc of mine who has a story now that he claims he can make certain nerve cells and suppress pain, at least in animal models, by transplantation. But again, it's early days for that. My feeling is the best role of stem cell is really modeling it. And you could really model it with your own genotype. Now, it could be your own stem cells, your own DNA. You could see where you respond to that drug in your genetic background. And so that's a very, very powerful way. And maybe just one more point is, don't make it too complicated, but for this drug testing, there's now the possibility to do it not just for one person at a time. Something we try to develop is when we model the disease in a dish, that we do what we call a village in a dish. a village of people. So let's say all of you in this room, I don't know what you have, 50, 60 people, I could make stem cells from all of you, and you could put all of them in the same petri dish and then give a drug. And we can see very quickly, so you have a technique where you can very quickly figure out which patient would respond to that drug and which one would not respond. So that's another very powerful approach is coming online where you can do that for hundreds, in the future maybe even for 1,000 cells. That could really change how you do drugs. Now you find out maybe all 20% of the people in this room, they respond to drug A, but 50% to drug B. And maybe you can actually predict it and give people the right drugs. So I think that's another very powerful tool that could be particularly helpful for things like treating pain. Yeah.
Thank you. That's an important question. So I see a question here. Yeah.
Hi, this is for Lorenz actually. So you just discussed about studying the brain in a dish, and I was wondering what you think of that idea, especially because the brain is so complex. So for the example of interneurons, there's so many different types and subtypes in populations with different roles. So I wanted to ask you about that.
Sure. No, that is obviously a very important question for two main reasons. Now, on the one hand, we want to model the complexity of the brain. Can you do it? And we are still not there, but you can get a little bit closer. For example, you mentioned this interneuron subtypes. We just published a paper actually about a month ago on making one of those subtypes, which is the latest born one. Usually these are cells that mature very, very late in life. And they are very important probably in neuropsychiatric diseases such as schizophrenia. And so we finally managed to make those cells, show how they are integrated in this mini structure. And what's cool is you can actually now see certain properties that are not just in an individual nerve cell, but these are interaction between nerve cells. So these are so-called, when you sleep, you have certain waves in the brain that occur. If you're active, you undergo cognitive tasks, or other waves, called gamma waves. And so you can actually see some of these gamma waves that are usually occurring under cognitive task. We can see in some of those, we call them organoids, brain-like organoids, when they have the interneurons, not if they don't have it. And so that allows you to get now pretty complex brain questions answered with that. It's still not far away from a brain. No, it just has some pieces of it, but it gets to the next level of complexity. But it raises then the second question, which is, are you going to get to the stage where some people think, oh, are you going to get so-called sentience? So suddenly your structure in this is going to start thinking. I think there's a very, very low or actually basically no chance for that to happen currently, because we know the brain is about 2-3 pounds. No, we would see in addition, a whole brain growing. And even during development, no, it's not completely clear now how much sentience you have. But it's an interesting question. Now, how far can you push it? And should there be any ethical guidelines? What if you take those structures, put them into monkey, and try to enhance the function of the monkey with a lot of human-derived nerve cells? Would that work? Actually, there was a big call from the organization ARPA-H from the government, where they wanted to make whole structures, whole cortical layers that they want to have for treating stroke and for other things. And so it gets more to the question, if you start replacing the cortex over time, it becomes an ethical question. No, should that be done? What happens to your thinking if you were to do that? So I think that a lot of ethical questions that could come with that, but we are still far away to really have that realized. But we're also not completely back to just having individual nerve cells. We can make pretty complex structures already. Yeah?
So interesting. Yes, please.
When you've introduced new stem cells, are they replicating inside the brain or the pancreas, wherever? And then do you end up with a colony of stem cells that are functioning properly, but you still have the original cells that are malfunctioning or performing? Are they then existing in parallel?
Either one, yeah.
I'm happy to try to take it, and obviously happy to hear what Sarah has to say. But yeah, so it's obviously depends what kind of treatment you're going to do. No, I have mentioned in the brain, in many regions, there's not much evidence that there are well-functioning stem cells there. But to your point, there is clearly a disease there. So even if you now replace or you add new neurons into a disease environment, would those neurons, the nerve cells, also get sick? Because the disease is still there. And we have a lot of data on this question, and it seems to be somewhere in between. For in Parkinson's disease, we have data from patients that got those fetal cells, died 25 years later. So the question is then, some of them died earlier, you get autopsy, you look at it, and it turns out the first 10 years, they actually seem to be resistant to the disease. They look still very young. They look basically as if they were again young, newborn or childlike cells that are fully functioning. But after about 10 to 20 years, they start getting some aspects of the disease, which they shouldn't get after 10 to 20 years. No, it should take 50, 60 years if it's a normal course. So we need to understand more about this interaction. But the other point is obviously for stem cells, in some cases, it's a very important question because if you, for example, in the brain replace the glial cells, which are dividing cells or can be dividing cells, could they then continue dividing? The cause of some of the glial tumors in adult brain, like glioblastoma, probably those glial precursors, oligodendrocytes and other cells. So they might not be true stem cells, but they are bad enough that they make a pretty bad tumor. And so that's obviously a big question. Now, should we do those treatments for cells that can continue dividing? And there are some groups that have put in new neural cells in the brain. And so it depends a bit on the organ system. In some cases, no, that's actually the goal. The best known stem cell therapy currently is blood stem cells. Blood stem cells you put in because every day they need to make millions, hundreds of millions of new cells. So they need to be able to proliferate. And if you replace them, fixing a genetic defect, that's going to be very important. But it's always the concern. I work at Sloan-Kettering, as most of you probably know, that's a cancer institution. So that's always kind of the yin and the yang. Could you cause cancer-like behavior in those grafted cells? But also conversely, can you actually understand cancer using those stem cell models? And we had some really intriguing work that we have done where you can really understand using the human stem cell models, for example, in a tumor called melanoma, know what are the key triggers, not only the oncogenes, but maybe cell-specific triggers that could contribute to tumor formation by modeling it in those cells, which would be very, very difficult to do in an actual person. And so I think, again, stem cells have this potential risk, and we have to be very, very careful, talk with regulatory agencies how to mitigate that risk, that they could maybe suddenly become aberrant when they are dividing. And on the other hand, there is a huge potential for repair and for modeling those diseases.
Great, thank you.
I mean, if you like me to add.
Yes, please.
Yeah, I was just thinking about the example of diabetes, where you still have your immune system potentially attacking any cells that you put in therapeutically. So I believe in those trials, they've kind of caged the stem cells that they're using to make, sort of making a new pancreas basically for the person so that the immune cells can't get in there. So your question is absolutely spot on, that the conditions that led to the disease in the first place are often still there. So we have to think about ways to kind of avoid that when we're using stem cell therapies.
Yeah, thank you. And so I think we have time for one more question. I see someone here with the mic.
I can address this to both of you, but it concerns the skin. And I know, Dr. Miller, that's one of your specialties. Do you deal at all with the vitiligo and the skin discoloration at all?
Yeah, vitiligo is very interesting, and actually it's a disease of the melanocytes, which Lorenz studies melanocytes, I guess, in other contexts. So again, it seems to be an autoimmune problem that attacks the immune cells when, they shouldn't normally attack your own cells, and they attack the melanocytes. So, again, whether that could be treated directly with, a stem cell therapy, I'm not sure, but it's certainly something that one could use stem cell derived platforms to study and to study that, the origin of what it is that causes your immune systems to turn on the melanocytes like that. I don't know if Lorenz like to comment, because
I think we're out of time. So, but you can definitely discuss it with Lorenz and Sarah at the reception downstairs. I want to thank you for being a wonderful and engaged audience, and especially thank Lorenz and Sarah for this discussion.